My name is Mac Durham. I'm going to talk to you today about phase contrast and DIC imaging. We're going to talk a bit about what these are used for, what the utility of these different types of imaging are, and then we're going to go through some preliminaries that will help you understand the theory behind them. So here are a couple of phase contrast images which are taken with different phase plates. And the bottom here is an intensity map, where the height of this map basically shows you the signal of these cells. And you can see you get quite a different image here. These use basically different phase plates. So the fundamental problem with a standard microscope is that cells are very difficult to see. That's because bright field microscopy is basically the approach which is used on most simple microscopes, and that basically relies on the cells absorbing more light than their surroundings. So more light passes through around the cell than through them, and that enables you to see the cell. So here are a couple different types of cells that we studied in the lab. You have eukaryotes, and we have bacterial cells. So eukaryotes make up things like plants and animals. They all have roughly similar cell types. They're on the order of 40 microns in diameter. Some are much larger. There's some plant cells which are much larger than this. And there's some which go down to about 1 micron. Bacterial cells are typically quite a bit smaller. These cells are about 5 microns long and about 800 nanometers wide. This is a species of bacteria we study in my group called Pseudomonas aeruginosa. And here they are crawling around on a surface. Generally, though, eukaryotic cells are larger than bacterial cells. You can find some outliers in both cases, but this is specifically what they look like on a bright field microscope. You can see you don't get very good contrast. You can't really see the cell relative to the background. We'll define contrast a bit more rigorously in a bit, but this cell here on the left is basically obtained by scraping some cells from inside your cheek. So the classical way to look at some cells from eukaryotes in the lab is basically to take a cotton swab, rub it around inside your mouth, and basically spread that onto a cover slip and put it onto the microscope. So they don't absorb much light because these cells are basically quite small and they're nearly transparent. So if they're thicker, they'd obviously absorb more light because of Beer's law. But these are indeed quite small, and most of them don't have any pigments. So things like phytoplankton cells, which are eukaryotes, they have things like chloroplasts. So plant cells, you'd be able to see them a lot better. Obviously, leaves and such are green or different colors depending on the time of the year you look at them. But you can see those pigments quite clearly in a microscope image, but without those pigments, you can't see much of anything at all. So one way that we typically increase the contrast is the stained cells. And this is a really powerful approach. It's really been pushed forward in the last few decades. So this is a classical stain called a gram stain. This will basically stain certain types of bacteria, known as gram-positive cells. They have a different type of material on the outside of the cells that make up the exterior of the cells than gram-negative cells. And this gram staining basically allows you to visualize them. So this pink bits here behind are human cells, as eukaryotes, and these bacteria have been counterstained so you can see them more clearly. So this is the old approach. This is the new cutting edge type stuff where they actually can label different parts of individual cells. So this is a fibroblast. This is the cell which basically produces collagen in the body. And it's been stained with 3 different stains. You have DAPI here, which is staining the nucleus of the cell. We have this stain, ActiStain 488, which is staining the actin fibers, the active filaments here. And these are anchored down by these things called viculin here on the edge of the cell. And that's been stained in red here. So we have blue, green, and red. Now, this viculin stain is what's known as an antibody-based stain. What they've done is they've basically taken viculin and injected it inside of a mouse in this case, and the mouse, the immune system of the mouse produces an antibody against this compound, and they can put a fluorescent dye on that and allow you to image where those bits of the cell are. So you can pay a company to do this. Actually, you can give a company a sample, and they'll inject it for animal for you and give you back the antibodies. So the upside of this is it allows you to stain certain cells. So in this case, we've only stained the gram cells. We haven't stained these human cells. And in this case, you've stained certain parts of the cell. The downside of this is it's really hard to prepare these samples. Obviously, this is quite advanced, making antibodies and such. This is typically done with someone that has a lot of experience and knows what they're doing. It's not something you can simply plug and play. The other big downside of this is it basically often requires you, staining cells often requires you to kill the cells. You can't do live cell imaging on using a lot of stains because those stains are lethal. So this really limits its use, and is a big push forward of late to try to understand what cells are doing in real time when they're exposed to different kind of complex stimuli. And obviously these approaches will not work for such experiments. So how do you boost contrast if you don't want to use stains? Well, this is a standard bright field image. These are red blood cells. We actually was reading through a textbook earlier, and it was saying that you should get students to poke their finger with a small needle to collect a small sample of blood so they can see these different types of images come up on the microscope. But I guess we can't do those kind of things these days. So this is a bright field image. You know the blood is relatively dark red because of the hemoglobin in these red blood cells. And these, you can obviously see these cells quite clearly in bright field. However, you look at these in phase contrast, it actually has a much better definition of the cells relative to that of the background. Now, how phase contrast, we'll talk about this, how it works in great detail in a bit, but basically these kind of microscopes, a phase contrast microscope transfers invisible phase shifts that are induced by the difference in refractive index of these cells relative to the media into visible changes in brightness or different pixel intensities on your camera. So this idea can be extended and actually used to measure variations in optical path length or potentially cell thickness if you have the same refractive index. We'll talk about this more. That's called quantitative phase imaging. We'll touch on that briefly. So you can see that already we have a lot higher contrast. We can also see that we have a bit of artifacts. These phase contrast images here always have a halo around the individual cells. And then the other technique we're going to study today or talk about today is DIC, or differential interference contrast. And this also measures differences in optical path length, but instead of comparing it against the background, it actually takes the derivative of the optical path length, and that's just a fancy way of saying it appears to have shadows. So if you look at this closely, you'll see that on one side of the cell it's dark, and the other side of the cell it's brighter, and it looks like kind of a mountain at sunrise where the light shine on one side of the mountain, but the other side is in the shadow. So I see, hopefully my image is not blocking any of these PowerPoint slides, but we want to basically go over a bit about what is light. So light is an electromagnetic radiation. We can see the entire spectrum of electromagnetic radiation and see where light fits into that. We have things like gamma rays on this side. We have radio and TV waves on this side. So visible light sits somewhere in the middle. It goes from around 400 nanometers to 700 nanometers. And the way we perceive light is based on, let's see if I shoot this over a little bit here. The way we perceive light is based on the cones in our eyes. So we have both rods and cones, and rods allow us to see things better in low light conditions. It gives us contrast, but these cones allow us to see. So rods are for more or less increasing contrast and whatnot, but the cones in your eyes are what allow you to see in color. And each one, you have a typical person has 3 different types of cones in their eyes. They have ones which pick up blue light, green light, and red light. You can see this is the light which which stimulates each one of these cones. So each of them have a different range, and your eye basically does something called spectral demixing. It allows you to a different wavelength of light will produce a different amount of stimulation in each one of these cones, and you can work out with your brain what color of light you're seeing. Now, I'm red-green colorblind. One of my cone types is not working too well. I can't distinguish some shades of green with brown, which is quite common among the male population, or relatively common. Some women actually have 4 cones, and this is said among some people to increase their ability to perceive different colors and distinguish them. So light can be, as you probably are aware, there's something called the wave-particle duality when it comes to studying light. We won't get into that, but light can be modeled as a wave. And we can all understand what a wave is. This is a typical toy, which you've probably come across before. And with a slinky, you can basically produce transverse and compressional waves depending on how you operate it. So operate a toy. So you can, if you play with it by moving it up and down, let's see if I get this in the right part of the camera, move it up and down like this, and tie the other end to, say, a doorknob, you can produce a transverse wave, these waves will propagate down the slinky. Some of them will reflect back towards you. You can also generate a compressional wave by gathering up a bit of the slinky and then releasing it, and that will basically allow a compressional wave to travel down the slinky. This is really, the compressional wave is analogous to what you'd have produced by sound. So sound is a compressional wave, where we're going to model light as a transverse wave with a certain amplitude, which causes a deviation that's orthogonal to its direction of propagation. Okay. So one way you can think about generating a wave is, again, tying this slinky, or let's say in this case, to make it a little bit less complex, it's just a string or a rope to a doorknob. And this girl here is basically moving her hand up and down. She's producing a transverse wave, and the string gets propagating down and to the end of the string. So we can define a few different properties of this this wave. First of all, there's a defined wavelength. That's going to be basically, if you look at a position or look along the x-axis here, which is along the direction of propagation, and we're looking at the amplitude here is in y. That is the transverse waves going up and down in y. Then the wavelength is going to be the distance between 2 peaks or 2 troughs, or 2 of a similar point anywhere on this wave. That's going to be called the wavelength. We're going to denote that as lambda. The amplitude, however, is going to be from the position of rest to a peak or to a trough. We're going to model this as a sine or a cosine. So the other property of the wave we need to think about is the speed of the wave. This wave is going to propagate along the axis here, along the x direction. And if you follow a point along this wave, it'll move along at a constant velocity v. So it has units of length over time, or in this case, meters over second. So this is called the phase velocity, but it's important to note that if you're vibrating a string up and down, if you painted a red dot, we'll say a white dot, since I have a red pointer here, you pointed a white dot on the string, that point would only move up and down. It's not propagating down the length of the string, it's simply going up and down only in the vertical direction. Now the other thing we need to think about is the frequency. So she's waving her arm up and down at a given rate, and the frequency of that's going to be basically 1 over the period. So if she's waving it up and down once every 2 seconds, it'll be the frequency will be 1 over 2, or 0.5 hertz. So this is the same plot showing at a fixed point along this rope the oscillation of this rope up and down. So here we have time axis, and the period can be measured as a distance between 2 peaks or 2 troughs. Okay, so we will we'll go and derive in a second how this phase velocity here relates to the wavelength and period here. But before that, we'll just note for a quick second that you can define this wave using either a sine or cosine. They're basically the same thing. They're only like sine and cosine are the same thing. Just they're out of phase by 90 degrees or pi over 2 radians. And you can write this down. The amplitude is going to basically scale this. We have a wave number, which is 2 pi over lambda, and a frequency, which is going to basically be 2 pi times f. It's the angular frequency, which is different than the frequency we just talked about. We'll go ahead and define this a bit more carefully, though. So if we write down our wave speed, we're going to denote that as v. So we have our wave speed is equal to some distance over time. And again, this is the phase velocity of the wave. It's not the how fast that white dot we painted on the rope moves down the rope. It's how fast let's say a crest or a peak propagates down the rope. That's going to be equal to v. We're going to denote that v. That's going to be simply given by lambda over t, where frequency f is simply equal to 1 over t, is the number of waves that pass by a fixed point per second. So therefore, you can simply rewrite this. V is equal to f times lambda. Okay. So, as noted in our slide a second ago, these are electromagnetic waves, right? So they both have an electric and a magnetic component. And we can talk about that briefly. So the electric field in one of these electromagnetic waves oscillates in a direction which is orthogonal to the magnetic field. So we can write that down using an equation like this. We're using sines and cosines as we did before. So we have, again, this wave number, which is 2 pi over lambda. We have an angular frequency, which is the same, similar to our frequency, but we've multiplied it by 2 pi. And the details here aren't super important, but I just want to note that there's both an electric and a magnetic field, which is produced by an electromagnetic wave, and they're basically orthogonal to one another. Now, we're going to define the electromagnetic wave by the E component, the electric component. Some older textbooks, when they talk about things like polarization, which we'll get to in a second, they define the polarization axis along the magnetic field oscillation direction. So we'll just to point out, basically, we're gonna stick with the current approach and go with this E variable. So that takes us to polarization. So many of you have probably familiar with polarization from sunglasses. We'll see an example of it in a second, but most light, when it's coming out of the sun or coming out of a light bulb or coming out of a lamp on the top of your microscope, it's unpolarized. That is, it's oscillating in all directions equally. So it has a direction of propagation. It's moving along this blue arrow, but it's composed of basically waves which are oscillating in all directions equally, and the directions which are orthogonal to that direction of propagation. So a polarizing filter basically absorbs all the light which is oscillating along a certain direction and lets light through, that's oscillating in a direction orthogonal to that. So if you have a natural incident light hitting a polarizer, it comes out of the polarizer with this, in this case, a vertical polarization. This wave's only oscillating in the vertical direction. The component of this light, which is oscillating in the horizontal direction, has been absorbed almost completely in this polarizer. This is called a Polaroid or polarizing filter. They both mean the same thing. So if you have unpolarized light which passes through a polarizing filter, it reduces in its intensity by 1/2. Okay, so we'll go quickly to this video, and you can see this is a typical, if you ever worked in the sunshine with a laptop, you've probably noticed sometimes you have a problem seeing your computer screen or your mobile phone screen. That's because the light from an LCD screen is polarized. And if you have, next time you're out with your sunglasses on, which have a polarizing filter in them, not all do, just the fancier sunglasses do. If you try cocking your head to one side, you'll notice that it basically becomes completely opaque. And that's because the polarized light, which is coming from your screen, and this orientation here, when it turns its hand sideways, basically these are cross polarizers, and it's blocking all the light. So to help understand what's happening in that case, if you have more than one polarizing filter, you have incident light here. This is, for example, from the sun. It's unpolarized, moving in all directions. It passes through this first polarizing filter, which basically takes this rope, which is moving in all directions around. It basically only lets through the vertical component, right? So this is similar to if you have a fence here with slats that are vertical. It only let through the vertical component, so this rope's moving around in a round direction. What comes out of this is going to be only something which is moving vertically. If you put a second vertical fence in front of it, then that wave will propagate through both of them. So after this filters out the horizontal components, this will be unaffected. This is similar to when you saw the LCD screen and the sunglass, and you could see light coming through both of them. However, you turn one of these fences the other way, or turn the polarizer. This is the equivalent to the case of what happens when you have this guy's turning his hand sideways, and you can't see any light coming through. You have one basically light coming through this LCD screen, which is polarized in one direction, and you turn the second polarizing filter here in a direction orthogonal to that, and basically you get nothing coming through. So you can see at the bottom here, no light passes through when these polarizing filters are oriented 90 degrees to one another. Okay, so with that, we'll go a little bit, do a little bit of theory here to cover what happens when light hits an object, which is a different refractive index. And we'll explain what that means in 1 second. Okay, so let's go on to a new page here. So first things first, when you have light moving through a vacuum, it moves at a really fast speed. So the speed of light, we denote that as c, so we're going to define that as speed of light when moving through a vacuum. That's going to be roughly equal to 3 times 10 to the 8 meters per second. So this is how fast light would travel through space, for example. It would travel slower on Earth because it has to pass through our atmosphere. So, but when light instead passes through an object, like it slows down slightly in air, but it slows down a lot more in things like glass or potentially through a cell. So when it passes through a material, light slows down And it slows down in a defined way, and we'll write down the speed of this light through this material as such. So this is the speed of the light, v, when it's moving through a material other than a vacuum, and n is something known as the refractive index. Okay. So we know the speed changes as it passes through material. What does that mean about the frequency in lambda? We know that we had this expression before. We had v equals the frequency times the wavelength. So if v changes, we know that either the frequency or lambda, f or lambda has to change. And I realized I misspelled wavelength there. So one of these has to change, or potentially both of them. But if you think about it for a second, you'll realize that the frequency cannot change. This is because an object, when light hits an object, the number of rays, so the number of peaks, for example, which hit this object and pass through it has to be constant. So these peaks, for example, it's a peak and a trough here, the different colors. You can't, the number of these peaks has to be conserved as it passes through this object. Peaks can't magically appear, disappear as it passes through this object here. This is called specimen in this case. So this has to be conserved. So if 10 wave fronts or 10 peaks hit it per second, there has to be 10 wave peaks coming out of this. These wave peaks can't be created or lost within the specimen. So therefore, the wavelength has to change. F stays the same, but the wavelength changes. So frequency does not change at the boundary between two different materials. And that's simply because the number of waves which enter the boundary has to be the same number of waves which leave the boundary per unit time. So because of this, therefore, lambda must reduce. And it must reduce in a certain way. So F is equal to, so F has to stay the same. We have the speed of light divided by the wavelength of light in a vacuum. And that's simply going to be equal to V over lambda, where this again is the speed of light through a material. And lambda here, without the zero, without the naught at the bottom, is going to be simply the wavelength of light in that material. Okay, and to note that we can rearrange the equation we had before. N is going to be simply equal to C over V. So then, if we note that, we'll just move into this little bit here. So, actually, let's go on to the next page. So it doesn't get cramped. So taking that, following on for that, so using those expressions we had on the previous page, so therefore lambda equals lambda naught times V over C, simply equal to lambda naught over N. So lambda naught is the wavelength of light in a vacuum, N is the refractive index of the material, and this here is simply equal to N. So this is smaller than 1, that bit in the dashed region, the dashed circle. Okay, so the long and short of this is that light travels slower through materials with larger N. Okay, and these 3 dots just simply mean therefore. So cells have a refractive index which is larger than that of 1. That is, light slows down when they go through cells. So these are different parts of a cell, and you can see that they have different refractive indices, and this difference in refractive index will allow DIC and phase contrast to make these different parts of the cell look different than the rest of it, because they basically rely on slowing light down. They increase the image by looking at differences in the rate at which light passes through these objects, like cells. So when light travels through these different objects, it basically causes what's known as a phase difference. And we'll get onto this and calculate what the phase difference is of light when it passes through a cell. But I think it's just important to note for a second that these different types of objects, which we can potentially image with a microscope, we can have something called an amplitude object. This would be something in which bright field would be very good at picking up. So an amplitude object will basically reduce the amplitude of light when it passes through it. So this gray dashed wave here was where the light would be, the wave would be if this object wasn't here. And you can see that the transmission of light through this object is less than 100%. So let's reduce the amplitude of this. Now we can also have a phase object, which actually lets all the light through. This is an idealized case. I think it's really rare that something would be let all the light through. But in this idealized case, it has an index of refraction which is larger than 1, and the light has slowed down. And you can see how this is this phase difference between this unperturbed light wave here, shown in gray, and the one which passes through this object. So then most objects have a combination of effects on the light which passes through it that are caused by both the phase and amplitude. And this causes a slowdown in the light, and it also reduces the amplitude. And so DIC and phase contrast obviously rely on the phase difference caused by such objects, and allow that to be converted into differences in amplitude at the imaging plane. So it's important to note that that if we'll just go back to our writing for 1 second, and just want to note that the intensity of light is proportional to the amplitude of the electric field wave squared. So if we have considering our electromagnetic wave. Just looking at the E component here. We draw that sine wave, or looks kind of like a sine wave. Let's draw it one more time. That's even worse. Okay, so that's a roughly okay sine wave. It has a certain speed v. This is going to be our maximum amplitude, E max. Or not the maximum amplitude, it is the amplitude. E max of this. The intensity of light caused by this is going to be denoted by I. And it's going to be proportional to E max squared. So intensity is proportional to the amplitude of electric field squared. So this means proportional to this symbol here. It's not exactly equal to it. There's some prefactors here which we're leaving out. But intensity, I'll just write down here. Intensity has units of watts per meter squared, or abbreviated W over m squared. So this is basically the amount of watts hitting a certain object divided by the area over which that wattage is spread over. So if you measure the intensity of any kind of light source, it'll be given with these units. Okay, so one thing we need to do next is just define contrast before we move on to the next bit here. And we said before that bright field microscopes have small contrast, but to define what that actually means, we can simply write down expression. So the percent contrast we call that C. It's going to be equal to IS minus IB over IB times 100%. And here simply IS is the intensity of the sample, or the intensity of a cell, for example, or a part of a cell. And IB is going to be the intensity of the background. Okay, so typically intensity is measured with a camera, and you can have different types of camera. There's different, you can get an 8-bit camera, for example, which will have 2 to the 8 power shades of gray, or 2 to the 8 power different levels of intensity. And under ideal conditions, this should be linearly proportional to the intensity. So what you would do in practice is you go and go back to our images of cells, and you could find, for example, what the intensity is at the center of one of these cells, this dark region, and this equation we just wrote down basically compares that to that of the background. So you can see that the difference between the intensity in the center of the cell and the region outside it would be larger for phase contrast than for bright field. So we would say that these phase contrast images offer better contrast. Okay, so we'll carry this on in the next video, which will talk about phase contrast.